And welcome to another session of our Sussex Vision Seminar Series, as always within the Worldwide Neuro Initiative. I'm George Kafedis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. As your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from Goethe University in Frankfurt, Professor Leo Peichl. For his bachelor's and master's, Leo studied physics at the universities of Stuttgart and Constance, and actually he was the first PhD student of Heinz Wessle. And during this time, Leo focused on the feline retina, characterizing mosaics and dendritic fields of different retina cell classes and separately on and off ganglion cells. He continued working with Wessle as a postdoc and in collaboration with Brian Boycott in King's College London. They contributed to uncovering the morphological and anatomical basis of retinal parallel processing. In 1981, he started his own lab in MPI for brain research in Frankfurt, and in 1987, he got his habilitation, if I'm pronouncing it correctly, becoming a professor at the Goethe University of Frankfurt, where he has been located ever since, and nowadays as a guest researcher in the medical school. Throughout his research years, Leo has retained an interest in the retinal circuits in a comparative approach and how their diversity reflects evolutionary processes or adaptations to lifestyle and habitat. Personally, I think that succinctly summarizing in this intro his work on different species is virtually impossible, but in a probably not exhaustive nutshell, Leo has studied retinas of birds, vipers, bats, rabbits, elephants, seals, wolves, horses, foxes, a whole diversity of rodents, lemurs, and marsupials. So in his big picture talk today, we have the pleasure of hearing about his work on a topic that has fascinated him since his doctoral years, namely retinal photoreceptor diversity across mammals. So without any further ado from my side, please all welcome Professor Peichl. Leo, the stage is officially all yours.
Okay, many thanks for the introduction, George. Very kind introduction, and also many thanks for inviting me to this renowned lecture series. So I feel honored, and I'm pleased to present you our findings, let's say, from over the last 3 decades or so. So it will be a rather big picture. Yes. Let me start the screen. So this, now let me get the pointer. Okay, here we are. Retinal photoreceptor diversity across mammals. And the reason that we're interested in that is that mammals have conquered all available habitat forms that exist on Earth. Most of them are land mammals, most of those living on the surface, but some also living below ground. Some have gone back into the water, like the seals and whales, and some, like the bats, have conquered the airspace. And the lifestyles also differ a lot. Some are diurnal, some crepuscular, some nocturnal, arrhythmic. This underground living ones live in a lightless environment. Some are herbivores, some frugivores, insectivores, carnivores, omnivores, what you have. And therefore, in some of them, vision is the dominant sense, but in other species, also other senses are dominant and vision plays less of a role. And we were interested in what consequences do these different lifestyles have for their photoreceptor properties? What adaptations and specializations are there? So basically, it's a question, how flexible is the basic blueprint of the retina as far as photoreceptors are concerned? And this, of course, is a zoological interest in the diversity of adaptive solutions, but it also is a paradigmatic interest in the general validity of the model systems that many of us study, preferably like the mouse retina or the primate retina. And let me say at the beginning, in this talk, I'll focus on the findings of our group and will only fleetingly mention the important work that other groups have done in this field. So please forgive me. Now, the outline of what I'm going to present to you is first general properties of the photoreceptors, the visual pigments, and what it means for color vision. So this is the rules, so to speak. And then in the second part, I'll show you some deviations of these rules or expectations that we had. One is ultraviolet vision. Another one is blue cone loss, particularly in marine mammals. And then at the end, I'll touch on subterranean mammals, where it was always thought that they have a very reduced visual system because in a lightless environment, you don't really need it, was the idea. Now, let's start with the primate retina, including our human retina. We have these different cell types in the different layers of the retina. And out here, away from the light which comes here from the lens and through the vitreous, we have the photoreceptors, which consist of two basic types, the rods and the cones. And what we usually do is we look on flat mounts of the retina to see the distribution of the cells and are able to determine the densities. And what you see here in a photo that I was given by Heinz Wessle from peripheral macaque retina, you see the cones in cross-section, the inner segments, which are labeled by a cone-specific marker. In this case, it's calcium-binding protein. And the unlabeled cobblestone-like pattern in between are the rods. And you see there are much more rods than cones. And that also holds for the human retina. So the cones, which are there for daylight vision and color vision, make up about 7 million in a human retina. And the rods are night vision photoreceptors, which don't contribute to color vision, make up about 120 millions in a human retina. So even us diurnal species have many more rods than cones. And this more or less holds for most mammals. So here is a comparison of mammals with different activity patterns. A very nocturnal species, the African giant rat. You see the cones labeled here, the outer segments, and everything in between is filled by rods, the unstained parts. And this species only has about 0.5% of cones. Then species like many carnivores, examples, the wolf here, which are nocturnal, but also tend to be crepuscular. They have a somewhat higher proportion of cones, about 2 to 4%. And that also holds for cats, dogs, etc. Then even more diurnal to crepuscular species like the sheep have still higher cone proportions, about 5 to 10% in the case of sheep. And here is the tree shrew, a very diurnal Southeast Asian species, which is very diurnal, and it belongs to the few mammals that have more cones than rods. And you see they are really densely packed, and about 95% of its photoreceptors are cones. And here you see the rods, which are really very sparse. So this is the exception among mammals. For example, squirrels, ground squirrels also, but tree squirrels too, have about 70% cones or so. So they belong to the few that have more cones than rods. So the basic take-home message from these comparisons is that first, all mammals have rods and cones, which is called the duplex retina, and the rod-cone ratio correlates with the dial activity. But most mammals still have rod-dominated retinae. even the diurnal ones. And that brings us to the first problem. So let us look at the rods first. When you look at cross-sections of the retina, a diurnal animal, like here the example of the squirrel monkey, has relatively many cones, the fatter, darker stained elements here, and a somewhat lower density of rods, which are the thinner processes in between. And therefore, the layer of their somata, the outer nuclear layer, is relatively thin to stack all the somata of those photoreceptors out here. Whereas in a nocturnal animal, example is the raccoon here, it has a much higher density of rods. And because the outer segments of the rods are very slender and thin, the larger somata have to be stacked in many layers down here. So the outer nuclear layer becomes much thicker. And that creates a problem because the light has to come through the whole retina to hit the light-sensitive outer segments of the photoreceptors. So the dilemma of nocturnal retina is because it has many rods, it has a thick outer nuclear layer and altogether is very thick. But thickness means light loss by scatter of the light while it travels through the very many membranes that exist here in between. So how did nature solve that dilemma? And I think we found out how it did that. So in a collaboration with a group from Munich, Irina Solovey and her colleagues, I'll give the names later, they are nuclear specialists. So they look at cell nuclei and they, by chance, came across the retina also and stained it with markers that are specific for euchromatin, the active part of the chromatin, and markers for heterochromatin, which is the inactive part that is not used by these cells. And you see that out here, the rod nuclei and the outer nuclear layer, they have a compacted central heterochromatin lump, and the active euchromatin is around in the periphery. Whereas in all other cells of the retina, and also actually in the cones, which are in between the many rods, the distribution is different. So you have the heterochromatin distributed also across the periphery and in different parts of the nucleus, and the euchromatin, the active chromatin, is much more prominent here. Which means these mouse rods, this is the example of a mouse, have an inverted nuclear pattern where the unused heterochromatin is in the center and the used euchromatin is in the periphery, whereas all other retinal cells, including the cones, have the conventional nuclei. And these are the members of that team with which I worked. And they came to me and asked me, is that generally so? It's a very unusual nuclear arrangement because it's not very good from a cell biological point of view. Then we joined up and included some biophysicists from Cambridge, Jochen Guck and Moritz Kreising, who were there at the time, and looked deeper into that. And did a check of many, about 40 different mammals from various orders of mammals and of various activity patterns. And indeed, in all nocturnal to crepuscular mammals, we saw this pattern with the central heterochromatin, the so-called inverted pattern. You see up here, this is a label for the heterochromatin, which is lumped, and this green is a label for the euchromatin, which is here around in the periphery, and this is the merge of the two images. Whereas in the diurnal mammals we looked at, the pattern is like you see in the other retinal neurons and in other neurons, other cells of the body, which is more distributed heterochromatin, more towards the periphery, and the euchromatin is more in the center, where it can make three-dimensional interactions between the different chromosomes. And when you push the euchromatin towards the periphery, the interaction is more or less limited to two-dimensional interactions, which is not good for the cell functioning. So we asked, why has that happened? And why is there this strange difference between nocturnal and diurnal rod nuclei? And when we order that in a phylogenetic tree, we see that there are, among the different mammalian orders, there are always a lot of nocturnal inverted nuclei, and sometimes then some of these species becomes diurnal and gets back to the heterochromatin or the euchromatin distribution that we regard as normal. So the most likely scenario is that early mammals were nocturnal and they invented this inverted pattern. And then readaptation to diurnality has occurred independently in several of the mammalian orders. So you see the Rodentia, then suddenly one species becomes diurnal. Up here, several become diurnal. Down here, the most of the primates are now diurnal, or many of the primates, sorry. But there are also still some nocturnal ones, like the lemurs. So this readaptation to diurnality during the evolution has happened in several mammalian groups. And whenever this happened, this return to diurnality, then the conventional pattern of the rod nuclei was reacquired. So this also means that it's probably better from a cell biological point of view to have this conventional pattern. And then we wondered, it has something to do with nocturnality, and perhaps it has something to do with improving vision. And so the biophysicists in the team measured the transmission of individual cells, individual dissociated rod nuclei from the mouse retina, and they found when it has this inverted pattern, then it acts like a little lens and focuses the light towards the outer segments in a more strong fashion than these conventional patterns, which more or less scatter the light. And then they modeled how a thick outer nuclear layer with rows of several rod nuclei would act on light that comes through the outer nuclear layer. And when you compose this outer nuclear layer of conventional nuclei, then you get a rather scattered image at the level of the outer segments. Whereas when they are inverted, then each one of them acts as a little lens, and the whole thing becomes like a light guide and produces a more focused and stronger signal. That means a better signal to noise ratio. So our conclusion was that these inverted rod nuclei of nocturnal mammals act as light guides, and that produces improved photon transmission to the outer segments. So it's an optical solution that sort of gives away some of the cellular properties, cell biological properties, to improve the optics. Now, okay, so much for the rods. Let's now get to the cones and to color vision. Again, we'll start with our human and old world primate color vision, which is trichromatic and is based on different opsins in the different cones. So we have red cones with a red sensitive opsin, green cones with a green sensitive opsin, and blue cones with a blue sensitive opsin. So each of these different cones contains its own opsin. And the spectral sensitivity of these opsins is shown down here. So we have a blue sensitive one, maximum in the blue, then a green sensitive one, and a red sensitive one, which actually has its maximum in the yellow to orange part, but its flank goes it's most sensitive, more sensitive than the other cones in the red part of the spectrum. You also see in black the rod profile, Stäbchen in German, which also is most sensitive in the green. So how did this come about during evolution? everything started with some kind of basic photoreceptor, probably long before the vertebrates appeared. And then about 400 million years ago, when the vertebrates appeared, there were 4 types of cones with 4 cone opsins and 1 rod type with a rod opsin, which is called rhodopsin 1. Then there is a shortwave sensitive cone opsin S1, which is usually ultraviolet. Then there is a second shortwave sensitive opsin S2, which is commonly blue sensitive. And then there is a green sensitive cone opsin because it was rather closely related molecularly to the rhodopsin. It was termed rhodopsin 2. And then there is a long wave sensitive opsin in the long wave cones, red sensitive. And that's true for most of the vertebrates still nowadays. And then during the evolution of mammals from sort of reptilian ancestors, something bad for us happened. It was about 150 million years ago. That is, the mammals lost these 2 opsin types and retained only 2, one shortwave sensitive, which is derived from this S1, but has been shifted towards the blue from the originally ultraviolet position. And the second opsin in mammals is a long wave sensitive, which derives from that long wave sensitive opsin phylogenetically, but has been shifted from a mostly red position in most mammals towards a green position, so that these 2 move a little more together. The rod has been kept. And then something happened during the evolution of primates, which was again good for us. And that was about 40 to 50 million years ago when the old world primates appeared. We kept the rods, we kept the blue cone, and there was a gene duplication in this long wave cone opsin gene. And the 2 copies diverged during further evolution by changes of a few amino acids. And now we have 2 copies of this long wave sensitive opsin in our genome, and one produces a green sensitive cone opsin and the other a red sensitive cone opsin. So we are trichromats on the basis of these 3 detectors. The majority of other mammals are dichromats on the basis of these 2 detector types, and most of the other vertebrates, the non-mammalian vertebrates, are tetrachromats on the basis of these 4 detectors. Actually, this is a very simplified scheme. I'll give you a few notes aside. In the reptiles, which usually have 4 cone types, most snakes only have 2 cone opsins and then lost the other 2. Among the birds, which are also mostly the diurnal birds, are usually tetrachromats. The nocturnal owls only have 3 cone opsins and some of them only 2. So there also is quite some variation. And then there is an exception among the mammals. The monotremes have retained the S2, the blue sensitive cone opsin, instead of the S1 opsin that we other mammals have kept. Okay, so how do we study this? This is sort of the summary of studies from many groups that have done genetic analysis of the opsin genes. And we look at it with histological methods with antibody staining. So there are antibodies specific for the blue cone opsin or the shortwave cone opsin that would label all these cones. And then there are other antibodies available that are specific for the long wave sensitive opsin, the green, or in the case of old world primates, it labels both the green and the red opsin because they are very closely related molecularly. And so what we get when we look at flat-mounted pieces of retina is this labeling of the different opsins in the cone outer segments. Here, the cat example, the long wave sensitive cones are labeled with the green fluorescent dye, and the shortwave sensitive cones with the magenta fluorescent dye. And you see the majority of the cones is green sensitive, and the minority is blue sensitive. That's also true for a very nocturnal animal like the rat. You have this mixture of a majority of green cones and a minority of blue cones. And the pig has a higher cone density, it's more diurnal, but again, these two types. And the tree shrew that you've seen with this high density of cones, it still only has these two cone types, many green sensitive and fewer blue sensitive ones. And this histological approach actually can tell you the densities of these different cone types and the mixture of the two cone types, which you don't get out of genetic analysis of the opsin genes. And so most mammals have two spectral cone types, the L and the S cones, which gives them dichromatic color vision. And commonly, the L cones form a 90% majority and the S cones about a 10% minority. And that's also true for animals that have for a long time been considered to have pure rod retinae and have no cones at all because they are so nocturnal. So when we looked in Microchiropteran retina or in Megachiropteran retina, so the flying foxes, and here these are the conventional bats that we have here, example of Carollia here, you see they also have these two cone types. And they have about 5% cones, so against the classical expectations that you find in older papers on the topic. And the same is true for the Megachiroptera, the flying foxes. They also have the two cone types. And they have somewhat lower cone percentages. But they belong also to this sort of normal mammalian pattern that has rods and cones in the retinae. So how does that look like? We old world primates see the whole rainbow with our three cone types. And the other mammals that only have two cone types, they can distinguish between short wavelengths, probably some kind of violet to blue to them, and the longer wavelengths, which are sort of depicted here as yellowish. But they cannot make the discrimination between red and green because they only have one cone type that covers this part of the spectrum. And at night, of course, we also see only in black and white because with one photoreceptor type only, the rods, you can't discriminate colors. This is an example of how this would look like in real life. I got this image from Carol Knights and Knights. They looked at the cone types, the spectral tuning of the horse cones, the two, and then took this picture. This is how we humans see it, and ran it through an algorithm with the two spectral cone types of the horse. And what you get is how the horse would see this image. So it can discriminate this sort of bluish violet colors from the others. But for example, what we see as a clear distinction between the red anorak sleeve and the green field in the background is the same long wave color for the horse. And if you happen to be a red-green blind human, you would probably see it more like this than like the upper image. It's also a bit out of focus, so to speak, because the visual acuity of the horse is lower than that of humans. Okay, so this brings us to the end of the first part, the rules or the basic pattern that we see in mammalian photoreceptors. All mammals have rods and cones, a duplex retina. Sorry. The rod to cone ratio correlates with the activity pattern. So nocturnal ones have higher cone, rod densities and lower rod cone densities. The diurnal ones, the reverse. And the rod nuclei of the nocturnal mammals is inverted, and the rod nuclei of the diurnal mammals are conventional in their nuclear architecture. We have two spectral cone types, which gives most of the mammals dichromatic color vision, with the L cones being most sensitive to green and yellow, and the S cones being most sensitive to the range of blue to violet. And L cones are a majority and S cones are a minority. But this is biology, and you know, biology always offers interesting deviations from the rules that teach us something about how flexible the system is. And now we come to these deviations from expectation. Now let's first look at this phylogenetic scheme again. We have our blue cones originating from ultraviolet sensitive S1 cones of the other vertebrates. And in the mammals, this has been shifted towards the blue. But some mammals have not done this shift. They have stuck to the ultraviolet sensitivity of their shortwave cones. So while the majority of mammals have their spectral sensitivities of the S cones in the wavelength between 400 and 450 nanometers, and the L cones around 550 or 500 to 550 nanometers. Some mammals have kept the ultraviolet sensitivity in the range of about 360 nanometers. So everything below 400 nanometers is ultraviolet. And the long wave cone opsin has also been moved a little to the short waves to keep the distance between these two and the overlap of the two curves more or less constant compared to the distance here. And this was first observed, this ultraviolet sensitive shortwave cones were first observed in the mouse rat and gerbil by the group of Jerry Jacobs, 1991 that was. Then we have found it in further rodents, the Dego, the Kururo, Tukutuko. We have seen it in voles. We've seen it in the European mole, which is an insectivore, and we have also seen it in the bats. They also have ultraviolet sensitive S cones. And it has also been found in some marsupials that their S cones are ultraviolet sensitive. So this apparently was not very difficult during evolution because the tuning difference between ultraviolet and violet and blue happens by changes in 3 crucial amino acids at these sites, 86, 93, and 97. So only a small mutation can produce a remarkable shift in spectral sensitivity. Nevertheless, they are also dichromats. So it's just a shift of the 2 existing cone opsins. And most of these UV-sensitive mammals are nocturnal. And that's probably because in the more diurnal mammals, the shift to the blue part of the spectrum is joined with an UV filter in the lens. So UV light doesn't even reach the retina, and therefore a UV sensitive cone wouldn't be of much use. So it was shifted to there. And the UV filter in the lens is probably there because high UV intensities damage tissue and therefore could damage the retina. So that's a protection against this UV damage. Whereas in nocturnal animals, light levels are generally lower, and therefore also UV levels, and that is reducing the risk of UV damage to the retina. And so perhaps these animals, these nocturnal animals, just didn't have to care where the spectral sensitivity is, and therefore left it where it was in the UV part. Now, there is one interesting animal, and that is the degu. The degu, you see it here, it's a relative to the guinea pig. It lives in South America, and it's very diurnal, and you see that at the cone densities that it has. It has about 30% cones, again, more green than shortwave cones, but the S cone spectral sensitivity is ultraviolet. We've measured that in collaboration with Chilean colleagues. They have done spectral electroretinograms to show this UV sensitivity. And then, of course, we were interested, why does this animal stick to the ultraviolet sensitivity, although of course, its lens has to be transparent, and it is to ultraviolet, and therefore, as a diurnal animal, it is at a higher risk for light damage, UV damage to its retina, and still it has kept the UV. So the Chilean colleagues went into the field and checked the potential UV signals in their habitat. And so they measured various substrates, soil and plants, etc., and found that there is very little reflectance in the ultraviolet part. So there is nothing that the animals might be very interested in and which reflects strongly ultraviolet. Then they checked the animals themselves, measured the reflectance of the back of the fur here towards the belly, and these animals have a rather wide belly. And indeed, the belly fur shows a higher reflectance in the ultraviolet than the back. And these animals, when they want to warn their compatriots from dangers from the air or wherever, they stand on their hind paws and show the belly to the others. So perhaps this is a signal that is enhanced when you can also see the UV part. You see also in another species, the Octodon bridgesi and the Octodon lunatus, it's also the belly has a higher reflectance in the ultraviolet part of the spectrum. But then what the colleagues also did was looking at the urine scent marks that these animals use to mark the territories and to give messages to their compatriots. And here, the surprise was that fresh Daegu urine has its maximum contrast and reflectivity in the UV part of the spectrum and drops above 400 nanometers. Whereas old dried-on urine has its maximal reflectance in the green part of the spectrum and very little in the UV part below 400 nanometers. So with these two cone types, an ultraviolet sensitive one and a green sensitive one, these animals can not only smell their urine scent marks, they can also see them and differentiate between freshly ones, which are of interest, and the old dried-on ones, which you don't have to care about. So the idea was that perhaps in such a Daegu terrain, they live in sort of subterranean burrows, but use connections between these different burrows a lot, so they have real roads there. These might be for a UV sensitive animal look like street illumination with all the urine marks along the mostly used paths. So they can see the hotspots where the action is going on. Okay, that's for ultraviolet vision in some mammals. Some mammals indeed are really colorblind. They completely lack the blue cones, have only the green cones, the L cones, which means they only see at daylight with one photoreceptor type, which means they see it in gray values, like we do at night with our one rod type. And the species that have been found to be lacking S cones, they have been studied by several authors across the world. One example is the owl monkey, a New World monkey, the bush baby, a prosimian. Some lemurs have been found to lack S cones, the raccoon and the kinkajou, which are carnivores. Some rodents, like the golden hamster, we have seen it in the beaver and in the fat dormouse. And we have also found it in some bats, microbats as well as megabats. So these are colorblind. They are all nocturnal, so you could argue it doesn't really matter because they look with their rods mostly anyway. Here, I'll show you the example for Madagascar lemurs that we have studied in more detail. So you see the whole spectrum of activities. There are diurnal lemurs, cathemeral lemurs, which means they are active at day and night. They are nocturnal. There are nocturnal lemurs like the mouse lemur, the smallest primate in the world, or the dwarf lemur. And their cone patterns also reflect these activity patterns. So relatively high cone densities in the diurnal Sifaka, again, the two types. In fact, it has been shown that this and also some of these cathemeral lemurs are trichromats. The females are trichromats because the L-opsin gene has a polymorphism. So the females with the two X chromosomes, where the gene is located on, can have two copies of the long wave sensitive cone. But generally, the males and genetically, they are all dichromats with these two cone types. The nocturnal mouse lemur has much lower cone densities, again, these two types. And this dwarf lemur is one of the examples who have no blue cones or no S cones, only green cones. So this is really colorblind. And now the mouse lemur shows you another example why histological assessment of the cone population is important and gives you more information than genetic analysis. So from a genetic point of view, you would say this is a dichromat. It has two cone opsin types. Now, when you look at the distribution across the retina, the L-opsin has its maximum density in the area centralis, the point of highest acuity in the mouse lemur, and the density drops off towards the periphery. Whereas the S-cone density has an inverted distribution. It has its highest densities in the periphery and around the central area. In the central part of the retina, there are very, very low S-cone densities between 0 and 5 per square millimeter. So most of the ganglion cells there would not be connected to an S-cone and would not get S-cone input. So with this topography, one can argue that the mouse lemurs are most likely colorblind in central retina, even though they have the basic set of two cone types. Okay. Now, the big surprise in this field of S-cone loss came when we started to look into marine mammals, the seals and sea lions, which are the Pinnipedia and the whales. And all the species we've looked at are lacking S-cones. That was a study done together with Ronald Krüger and Günther Beermann. So the consequence of that is the pinnipeds, the seals and sea lions, are offspring of terrestrial carnivores that went back into the sea. The whales are offspring from artiodactyls, even-hoofed animals that are terrestrial, and they also went back to the sea. So they have very different origins, but their body shape has become very similar as a convergent adaptation to this marine habitat. And in both cases, these terrestrial relatives of them have S-opsin, and the marine members of this large group have all lost their S-opsin. We did this again with histology, as you might imagine. So we compare here sheep as a terrestrial artiodactyl with the pilot whale as a marine artiodactyl subgroup, as you like. They have L cones, but these marine species don't have the S cones that you find in the terrestrial artiodactyls. Same is true for the seals. The wolf as a terrestrial carnivore, seal as a marine carnivore, both have L cones, but the marine carnivore, the marine seal, seals, all of them do not have the S cones. So that means they only have one cone type. They can't discriminate between colors. They see the world in gray tones, and that means for them the ocean is not blue. They are L-cone monochromats, most likely colorblind, and certainly they have a low sensitivity for the blue parts of the spectrum with the lack of S cones, which is a paradox because in clear ocean water, the blue parts of the spectrum penetrate deepest. This is shown here in this diagram. So water depth here in open ocean, and the sun hits the surface with its full rainbow spectrum, and the deeper the light penetrates, the more scattering there is of the very short and the very long wavelengths. So at the end, the deeper you go, the more blue the whole environment becomes. Now you could argue in a monochrome blue environment, color vision is not much use, so you can kick out one cone type. But I, in that situation, would have kept the blue cone, and they didn't. And that, of course, poses a question, why did that happen? And perhaps the solution is when you look at the light distribution in coastal waters, because here, first, you don't get so deep with your light because it's more turbid. And second, the longer wavelengths around the green to yellow part of the spectrum penetrate deepest, and that's because there are a lot of substances floating around in coastal waters. There are little sand grains and things that have been floated in by the rivers. There are also debris of coastal aquatic plants like kelp, etc. And that all leads to a stronger filtering out of other wavelengths and keeps the middle wavelengths penetrating deepest. So, and as whales and seals derived from terrestrial ancestors, in their early phase of going back into an aquatic environment, they were amphibious. So they stayed partly on land, partly in water, and certainly mostly inhabited coastal waters. And perhaps in this situation, where you have not much of a spectrum, so you cannot do color vision anyway, they have kicked out the blue cone, which was of little use here, and have kept the long wave sensitive, green sensitive cone. And now for the seals, for example, which are still more coastal and go on sandbanks, etc., so they are still rather amphibian, that might still be a very good solution. Whereas the whales that went later on out into the open sea and suddenly hit on a different spectral distribution of their waters might have now wanted their S cones back. But of course, once they have been removed, they can't be gained back. Okay, so this is our hypothesis. It's pure hypothesis about why the marine mammals have lost their S cones. Now, I'm already over time. George, is it okay? A few more minutes?
Yeah, absolutely.
Okay, thanks. So in fact, this is studies from other colleagues. Some baleen whales have actually lost both cone opsins, and they are rod monochromats. Those are the deep diving baleen whales. Perhaps they had to really focus on rod vision and get rid of the cones. But the cones remained as cellular entities. So the opsins have disappeared, but the cones as cells are still there. And cone bipolar cells are also still there. And that's important because in a mammalian retina, all the rod signals are fed into rod bipolar cells, and these rod bipolar cells do not make direct contacts with the ganglion cells. They feed their signals through A2 amacrine cells into the cone bipolar cells in order to get to the ganglion cells. So losing the cones altogether, and therefore also the cone bipolar cells, would have been really detrimental for the vision altogether, also for the rod vision. On the other hand, we've also looked at other aquatic mammals or semi-aquatic mammals, and all of these still have their S cones and hence cone dichromacy. So there is the manatee that has been shown by other colleagues that it has S cones and L cones. We have looked at the hippopotamus, which had them, the river otter, the sea otter, and the polar bear. So this loss of S cones is really a speciality of the seals and of the whales. There is no sort of general pressure. Being aquatic means you have to get rid of your S cones. Now, the final topic I want to briefly touch is subterranean species, which have been sort of postulated that they have very reduced visual systems because they don't need them, and some of their eyes are even hidden under the fur, closed lids, like in the spalax. But 300 subterranean species have evolved independently among marsupials, insectivores, and rodents. So from surface-dwelling ancestors, they have moved underground, probably for safety reasons and for a more stable, not so much changing environment. And so the question is, are they really as blind as mole? We have looked at various of these, some from South America, some from Africa, and at the European mole. And the example which is perhaps most telling is that of the rodent family Octodon, where we have already seen the degu as a diurnal octodon. And there are these nocturnal relatives, Octodon britgesi and Octodon lunatus, which are both nocturnal. They are surface dwellers. And then among the octodons, there is one diurnal, one subterranean species, the Kururo, or Spalacopus cyanus. Subterranean, lives and forages underground, and has very rare above ground activity. So here one can directly compare within one family what retinal adaptations to the subterranean environment are. And we have seen the high cone density of the diurnal degu, about 30% cones. The nocturnal members, here the example of Octodon britgesi, they only have 2% cones. So densities are here 55,000 per square millimeter, here only 10,000 per square millimeter. Typical nocturnal situation, lower cone densities and higher rod densities. Now the Kururo, the subterranean species, has an interestingly high cone densities. It has about 10% cones and up to about 30,000 per square millimeter. So much higher than this nocturnal species. And this was really a surprise to us. And the same turned up when we looked at the European mole. This is a flat-mounted mole retina, rather small, about 1 millimeter or so in diameter. But it has these two cone types, the shortwave sensitive ones and the longwave sensitive ones. Typical mammalian situation, and again, cone proportions of around 10%, 9 to 15%. So much higher than you would find in a nocturnal surface living mammal. And also we found that the S cones are UV sensitive. So that is this example. Same is true for the mole rats, these African mole rats. Sections through the retina, cone transducing, labeling all the cones up down to their pedicles, and the S-opsin co-labels most of these cones. So most of these cones contain the S-opsin. Again, this animal has about 10% cones, very high cone proportion. Here is a double labeling of the S-opsin and the L-opsin on a flat-mounted piece of mole rat retina. And you see there are more S-containing cones than L-containing cones. And in fact, many of them are dual pigment cones. That means they contain both cone pigments. And only a very small minority are pure L cones, and a somewhat larger majority are pure S cones. So 90% of the cones here dominantly express the S-opsin, either exclusively or dominantly. And this dual pigment cones is also something that happens in quite a number of mammals, that cones co-express this. And the mouse is the most well-known example, where in the ventral mouse retina, most of the cones express both opsins simultaneously. And this high cone proportion in these subterranean mammals comes from the fact that their rod densities are reduced, and therefore a sort of a relatively normal cone density would make up a higher proportion. So, for example, in a mouse, you have about 450,000 rods per square millimeter, whereas in the Kororo, you only have about half of that, 220,000, and the mole rat, only about 100,000 rods per square millimeter. So relatively high cone proportions and relatively low rod densities seems to be something very common among subterranean mammals. And this, of course, challenges the general view that there is a convergent adaptive eye reduction and loss of vision in subterranean mammals, which is the sort of classical view. And we think that perhaps subterranean mammals have a basic rod-cone mix, sort of 90% rods, 10% cones. And when neither the rods nor the cones have been under adaptive pressure, this basic mix is just maintained. It's useful when you happen to come above ground, your cones might be used. When you are underground, there is no light, and therefore you don't have to invest into improving rod vision. So you can just stick to that basic rod cone mix. Okay, so this is it. Sorry, I've really taken a long time. Let me just briefly summarize. All mammals, or now I have to say nearly, because these baleen whales do not have cones. So nearly all mammals have rods and cones, and therefore a duplex retina. Nocturnal mammals have this unique inverted rod nuclear architecture that where these nuclei act as light guides. Most mammals have these two spectral cone types, and therefore dichromatic color vision, but some have only the L cones, and therefore cone monochromacy or complete color blindness. In most species, the S-opsin is tuned to blue or violet, and in some species to UV. And in some species, example of the mole rats, many cones co-express the L and the S-opsin, and are therefore not very useful for color vision anymore. And then many subterranean mammals have a rather normal set of photoreceptors, so there is no apparent regression in many of them. And that really is what I wanted to tell you. Here are my colleagues that I acknowledge for their contributions to these different studies. You can look, they are spread across the world. And without their help, all these studies would not really have been possible. So thank you for your attention that I have really stressed a bit. And that's it from my side.
Thank you so much, Leo, for this wonderful, far-reaching presentation in terms of animal studied. I'm both impressed and slightly jealous, to be honest. I'm just stopping your screen sharing just so we appear bigger on people's screens. I'm already posting the Zoom room link in case someone would like already to join us, because as a reminder to the audience, after this first round of questions that I moderate, we will be continuing briefly for a couple of minutes for offline discussions or greetings and whatnot. So the first question that I have personally, given how many different animals you have studied, is if you ever attempted to stain for non-visual opsins, because for example, what I think is pinopsin, when it was first described, was in chicken in the pineal organ and was not detected in the retina. So they are like, okay, non-visual. But then it turns out that frogs, teleosts, and sharks express it in the retina. So I was wondering whether you ever tried different stainings in these retinas.
Yeah, no, we haven't done that. We have done a few attempts at melanopsin, but nothing that would sort of show anything different from what has been published. So we stuck to the classical opsins.
Yeah, and it makes sense, to be honest with you. Returning to the chat for questions, one is from Tom. So it goes back to the heterochromatin configuration, this inverted pattern. And he asks if you would expect this pattern, like if there are examples of retinas where the inner nuclear cell body, not the outer, has this segregation pattern.
We have no examples for that, but we have only looked in mammals. So when you look outside in other vertebrates, well, there might be surprises. I don't know. But apparently, the mammals have stuck to changing the outer nuclear layer rod photoreceptors in that manner.
Right. And if I may add a sub question to that, like you mentioned that this inverted pattern was kind of invented in mammals. Do we know for a fact that it doesn't exist outside of mammals?
No, not really. We have started together with the Munich colleagues, Irina Solovey and Boris Joffe, to look into birds, compare nocturnal birds and diurnal birds. But there the pattern was so different that we haven't really followed that up. So it was hard to interpret. What I can say is that the monotremes, the platypus and the echidna, have a conventional organization, even though they are partly nocturnal. And this inverted nuclear arrangement obviously has only been inverted after the monotreme split off the marsupials and eutherians. So we find it in placental mammals and in marsupials, correlated with diurnal versus nocturnal activity, but not in monotremes.
And this you have found only rods, if I understood correctly, right?
Yes.
And this is interesting because I think like there was a paper from Whaley 2 or 3 years ago showing that mitochondria in the cone-dominated retinas of ground squirrels, they act as optical fibers, like focusing the light on cones, right?
Yes.
Together
Yeah.
with the liquid, with the lipid droplets you have in chicken, where again it is for cones probably.
Yeah.
So it seems that you try to achieve the same end result, but with different means in rods.
Right.
Or
Yeah.
cloning.
That actually these mega mitochondria also exist in the tupaya, the tree shrew, which has this very high cone density. There it has also been described. So that's another sort of way of implementing some kind of optical focusing. Yes.
Right. Thank you very much for addressing this. Another question appearing in the chat is again from Tom for the Dagu that you mentioned. Has anyone done any behavioral experiments to see if their efficiency of warning each other is dependent on the presence of UV light?
Not as far as I know. It has not been done. But perhaps I'm not up to date with the whole behavioral literature on Dagus.
Right. Okay, I think these are the questions that currently appear in the chat. There are, of course, a lot of greetings and thank you for you, but you will see this, they will remain available after the talk.
Yeah.
I'm posting again the Zoom room link, and I will continue with one last question from my end if there are no more appearing in the chat, and then we can continue offline if there is interest. So my last question goes to the last part of your talk, or like semi-last, where you talked about the marine mammals and convergent evolution, where they lost S cones. And this is already the case in sharks, for example, right? So they also don't have S cones at all, let alone some don't even have cones.
Yeah.
And I was wondering whether this is not a matter of convergent evolution on the visual systems, if it is more like a constraint that you try to get rid of, because maybe other sensory systems are more dominant in that environment, be it, electrosensory or olfaction and stuff like that.
Could be, because some of the whales are echolocating, so they need some space in the brain to process that information. Certainly, when you lose a cone type and therefore color vision, processing capacity in the brain for vision can be somewhat reduced, and you can free that for other jobs. But that's speculation.
Right. Yeah, of course. I mean, I was asking for this kind of
yes.
answer. Okay, lovely. So given that there are no more questions appearing in the chat, I think it's time to take it offline. So at this point, thank you so much for this wonderful talk, Leo.
Thank you for having me.
And we're officially offline. I don't know